A principal advantage of our technique is that it allows the simultaneous recording of gaze position, pupil size, and single neurons in patients implanted with depth electrodes at the bedside. This allows us to determine whether there are neurons in the human brain whose activity is sensitive to eye movements and to the identity of the currently fixated stimulus.
While this technique is not designed to study a particular disease, it is well suited to study neurological diseases that result in abnormal eye movements or other oculomotor abnormalities.
Demonstrating the procedure will be Nand, Chandravadia, and James Lee, research associates from my laboratory, and Erika Kwan, a neurodiagnostic technician at Cedars-Sinai.
To begin, connect the electrodes to the recording system and position the patient sitting upright. Then, connect the stimulus computer to the electrophysiology system and eye tracker. Place the remote non-invasive infrared eye tracking system on a robust mobile cart. Then, attach a flexible arm that holds an LCD display to the cart. Place a fully charged uninterrupted power supply on the eye tracking cart and connect all devices related to eye tracking to the power supply rather than to an external power source. Make sure the IV device connected to the patient is running on battery and is not plugged into the wall. Start the